Welcome back. We are still discussing about the future direction nanobiophotonics in particular and biophotonics in general will take, and thus far we have discussed about biomaterials, trying to explore it a bit more. So, in today's lecture let us discuss about bio-derived materials, materials which are derived directly from biology. So, I have discussed a bit in the previous lecture. Let us see what are the advantages of such bio-derived materials. Bio-derived materials are obviously naturally biocompatible, making them suitable for use in biological and medical applications. Bio-derived materials can be engineered. You can engineer them, you can artificially create, you can convert them, you can modify them. Bio-derived materials can interact with other biological molecules and create some sort of a chain reaction, modify them without completely destroying them. Natural selection found in biological material can be mimicked to create photonic device with unique optical effect. So, biological system, organic systems evolve, they evolve and there is a natural selection, there is a natural bias in the different organism that comes out, basically the so called survival of the fittest, and natural selection comes into. So, natural structures found in biological materials, they can also produce some kind of a bias towards the strongest structure available. So, the evolutionary principles can also be brought in. Of course, natural structures in biological materials can be mimicked and bio-derived materials often have lower carbon footprint, basically eco-friendly, but fascinating aspects can come up. You derive 3 or 4 biologically inspired materials and then you find out that their interaction with light or their interaction with other biological materials, there is a preference for one particular bio-derived material versus the other. So, here are some examples: bacteriorhodopsin for holographic memory, GFP for photosensitization, DNA as host of laser dyes and biocolloids. We discussed this, but let's go little bit detail. So, bacteriorhodopsin is a light absorbing protein found in the purple membrane of certain salt loving bacteria, particularly Halobacterium. This protein has gained significant attention due to its unique photochemical properties and potential application in various fields. We have discussed them in brain based imaging and whatnot. But these days they are using it for holography, they are using it for holography, light based memory sensing. Bacteriorhodopsin's chromophore is a molecule called retinal, also used in our eye for visualization. Retinal is covalently linked to the protein and changes its configuration upon absorption of light. This change in configuration triggers a series of events leading to proton pumping. Now, think about it. A particular material covered with retinal or covered with bacteriorhodopsin. It is connected with particular protein. So, bacteria or retinal part of bacteriorhodopsin connected with a protein. As soon as it absorbs light, it kicks the protein, change its isomorphism from cis to trans. So, you have a thin film made up of this particular chemical retinal connected with molecule, and you have then hit specific area of the thin film with specific laser, and only those areas where laser has hit has a isomorphism, a change in conformation of the protein, rest of the area it does not, right. So, those areas where it has illuminated, say you call it 1, those areas where it has not illuminated, call it 0. So, in a thin film, small area, you can write 1 0 0 1 1 1 etc. Can they be served, this bit stream, into some sort of an information, some sort of a memory, and after some time you just wipe away everything, you just put some kind of a potential, an electric field, or shine some other kind of light so that it resets. Resetting is quite easy, you reset and then redo this process. So, is this some kind of a rudimentary memory that you are trying to get in a small area in a biological membrane? Remember, this is biological, this is not silicon out of which you will make a flip flop or a logic gate. This is purely biological, and you are writing the memory by light, and this light will come from a laser which has been created by DNA. How cool will that be? The particular part of your body, think about it, could be used to store information, biological information. Several advantages of bacteriorhodopsin in photonics. It's robust and stable. It's naturally occurring protein that can withstand multiple cycles of excitation and relaxation without significant degradation. It's easy processing. It can be processed into high quality optical films, making it suitable for various photonic application. It's flexible. Bacteriorhodopsins can be chemically and genetically modified, allowing it for tailoring of different types of light hitting, producing different types of isomorphism. High quantum yield, bacteriorhodopsin exhibits very high efficiency. So, how much photon results in how much amount of isomorphism, the efficiency is pretty high. Photo conversion between different states and tunable photophysics. It undergoes a well defined photochemical reactions leading to changes in absorption and refractive index, as I was saying, which can be exploited for making photonic devices such as hologram, such as light based memory, such as photonic crystal, frequency filters, optical frequency filter and what not. We have multiple photonics applications of them. We can have random access RAM, basically random access memory, thin film memories, photon counters and photovoltaic converters. How many amount of photons have been hitting this particular area, right? How much amount of photon hits in your retina for it to convert a particular message that you see, the total number of photons required. You can do all these kinds of fundamental calculation, because the number of photons is directly proportional to the number of isomorphism that has happened. The protein has changed its morphology. So, by looking at the amount of morphology change, you can back calculate the amount of photons it has received. It is reversible, yeah, artificial retinas, that is what I was saying. People who have by glaucoma or some other kind of retinal based disease who have got blinded, can it be utilized. And of course, these days it is quite popular to have pattern recognition system. A particular biophysical, photophysical, photobiological activity has a particular amount of photons, particular frequency, particular time interval, how periodic it is, how different it is. If we can arrange, we can understand the pattern, we recognize the pattern, of course using machine learning and what not. You can have a fundamental knowledge, of how the entire process has taken place. This is a laser Doppler holography of blood flow, one behind me, in the optic nerve head region of human retina. So, this is just to see the blood flow in the optic nerve, right. Of course, GFP, I do not think I need to discuss this in detail. We have discussed GFP in detail previously. Is a naturally occurring protein, have a strong green fluorescence when exposed to UV light, ultraviolet. It was discovered in jellyfish, and GFP's ability to emit green light upon illumination has made it an invaluable tool, especially in fluorescence. You are not using any dye, you are simply coding GFP as a gene in some kind of a animal and trying to see it. There are several advantages of GFP, there are different variants, although GFP stands for green fluorescent protein, but nowadays you can have blue, cyan, I do not know what cyan color is, yellow and red fluorescent proteins. For us it is red, blue and green, cyan and then mauve and purple. These I hear from my daughter and I have no idea what that means. Fluorescent markers, GFP can be used for FRET, Foster resonance energy transfer, non-invasive imaging, targeted labeling and quantitative analysis. We can utilized for protein expression level, concentration and interaction. There are several photonic application, absorptions of UV and visible regions suitable for photosensitization, presence of neutral and anionic resonant forms the chromophores. GFP structural stability insulates the chromophore from the environment, like oxygen quenching. This quenching is a huge problem in fluorescence, GFP reversible denaturation and renaturation properties often control over it optical properties. So, you can utilize it for fantastic fluorescence imaging in vivo in a dynamic process. You do not have to kill the cell and then observe it, it can evolve. You put some amount of this GFP either encoded into a living organism and then the organism grows and you constantly see the changing function of a particular set of proteins, particular set of organ, particular set of tissue, a cellular structure, morphology, etc., all changing with respect to time in a live tissue. You can have a live image from a molecular standpoint. So, of course, DNA in photonics, this is also something that is coming up, though not 100% that developed. DNA is the fundamental genetic material and has shown promises in various photonic application. Researchers from several institutes have naturally occurring DNA to form templates, add several of them together and make thin films which can have unique electro-optic property or optical property, where a particular wavelength of light is allowed to pass through, rest of them are all discarded. See how beautifully naturally occurring nanostructure as well as periodic structures these are. So, only specific photons, specific energy of photons will be allowed to pass through them without getting destructively interfered, and they could be utilized for several purposes such as making waveguides and optical films. DNA can process into high quality films suitable for waveguiding and light propagation. As I was saying, the inherent structural morph properties of DNA can be harnessed to create thin films that guide light, making them potential component of optical circuits. Fluorescence dye interaction, DNA double helix structure produce a platform for intercalating fluorescence dyes. Fluorescence dyes can be bind to the space between DNA base pairs, leading to enhanced. So, between base pairs you can have fluorescent dyes just in between these. Enhanced fluorescence and potential application in sensor and imaging. Amplified spontaneous emission, this is where the laser is coming from. DNA films doped with fluorescence dyes can exhibit amplified spontaneous emission, where light amplification occurs due to stimulated emission. You know it in the other way, light amplification by stimulated emission of radiation, LASER. The property is essentially for constructing lasers and other light amplification devices. Application of DNA in photonics, nonlinear optical effect, we discussed nonlinearity, photochromic and nonlinear optical dyes. DNA can host photochromic and nonlinear optical molecules. These interaction often open avenues for designing switchable and tunable photonic devices. Biosensors are always there. A single strand of DNA can be utilized as a sensor which will only attach covalently attached with its complementary pair. If a particular virus has the complementary pair, you are able to detect it. If it is not attaching, then that particular virus is not present, DNA based virus. Similar thing can be done with RNAs as well. DNA based photonic devices, researchers have proposed DNA based materials for various photonic devices like switches, modulators and sensors. So, DNA based photonic devices, this seventh part is something that is really cutting edge. So, see, there is a natural tendency among electronics engineers as well as this photonic engineer these days is to convert from silicon to biological materials and especially utilize them as some sort of an implant into human bodies. You already have implant pacemaker, this, that, but can we make it more and more biocompatible, or in other words create a biological material that act as switches, modulator, sensor. A part of DNA in my body is acting as a sensor, glucose sensors, and if it is it sees some sort of a disturbance, it alerts me somehow. Is it possible in the near future? Think about this. Unique bioobject for photonics, this is what I was telling you. You have seen this, probably images of this. This exists inside sea, yellow tube sponge, but many people keep this as a handicraft or decoration in their houses. I have seen, one of my neighbor used to have this. You might have seen this, but what I am telling you is that these can be used as some sort of a template, right? They have a periodic structure. They have a periodic structure. This is also some sort of a periodic structure. Either you can use it as a template or utilize this and send light into them. The light will get diffracted or interfered, and that light could be utilized differently. Nature offers bioobjects like viruses, sponge, sea urchins, needles, etc. that has various periodical nanostructures of various dimensions. This bioobject possesses heterogeneous and precise surface chemistry, making them potential building block. These bioobjects offer the advantages of precise structural and surface chemistries, manipulating the refractive indexes and produces unique optical property. So, it has a unique periodical change in refractive index. This also a periodical change in refractive index. If the light is coming from this will have, the red part will have a different refractive index, the ash color part will have a different, and again the red again different. So, relative change in refractive index, this is the basic idea of photonic crystal. This is exactly what is a lattice of a semiconductor is. The lattice of a semiconductor, an electron visiting a lattice of a semiconductor undergoes a change in periodic potential. The opposite is a photon visiting a nanostructure, a periodic structure, a periodic photonic crystal structure undergoes a change in refractive index, or we call it dielectric constant. N1, N2, N1, N2, N1, N2. This is the refractive index N1, this is the refractive index N2. The inside is the refractive index N1, outside is the refractive index, the one which is the up part, refractive index N2. So, constant variation of N1 and N2 is equivalent to constant variation what the electron gets when it goes through a lattice, periodic potential resulting in formation of band gaps, valence band and conduction band. In this particular case, the periodic refractive index change, dielectric constant change of photon encounters results in formation of photonic band gaps. So, they could be utilized. It can be used for making naturally occurring photonic crystals, traditional colloidal crystals used for photonic crystals. Opals are by definition 3-dimensional photonic crystal. People use opals as some kind of precious stone for some kind of a ring, or even you will see it in their necklaces. There are iridovirus photonic crystals. The capsid was used to create photonic crystal, and you manipulate basically refractive indices and create frequency filter devices, modulator, switches, etc., etc. So, in conclusion, bioderived materials leverage the unique properties of natural substances, offer a rich source of inspiration for photonic applications. Biomolecules like bacterial rhodopsin and DNA exhibit intricate structures and behavior that can be harnessed for photonics. These materials align with sustainable and eco-friendly approaches, and they are often biodegradable, making them suitable for biomedical applications. So, these are the concepts that I covered today, and this was my reference as always, and I will see you in the next class. Thank you very much.